Welcome, grade 12s of South Africa. Now we're going to go through genetics as well. You've done the terminologies. Now we move on to Mendel's laws. Now they're not hard, and the words in the law are self-explanatory, and we'll get to them. But first we have to look at the history of genetics. Now, there was a chap called Gregor Mendel, and he lived in 1822 to 1884. He was an Augustinian monk, and he was a teacher, enjoyed experimenting with plants and investigating the outcome. Now, people, he was the first biogeneticist in history. So if you think about it, the laws that this man wrote in it would have been about 1860, apply today. He wrote the first laws, and we use them in genetics today. And genetics, by the way, if you enjoy this, is a very, very interesting subject, and it's a wonderful field to go into or to consider going into if you want to go into any kind of medical field. You have all kinds of calculations. You have to be very organized, and you have to have a very logical brain. But your basic principles that you would do in genetics is what we are going to learn here. So remember, grade 12 is very basic. It's pretty much the tip of the iceberg, but a very, very interesting subject nevertheless. All right, so Gregor Mendel, he studied the character of garden peas, and when we get to his laws, they revolve around those garden peas. He also wrote the laws that based on all his findings and are used in genetics today, which we've already said. Now, there are 4 types of actual genetic crosses, and if you know these 4 basic types of genetic crosses, then everything else is a piece of cake. There really only are 4. So if we look at those 4, which we've done, but we'll go through again, is the following. You are going to have, and I'm just doing a very basic, I'm not putting in genes and gametes and meiosis and all the rest of it, but these are your 4 basic crosses that you have. There's the Punnett square. We're going to go homozygous dominant versus homozygous recessive. Okay, here all your offspring are going to be heterozygous. So that would be number 1. Number 2, the second type of cross that you're going to get, you're going to take heterozygous versus homozygous recessive. So capital B, small b, small b, small b, capital B, small b, and small b, small b. So here you had 100% heterozygous. Here you have 50% would be heterozygous, and 50% would be homozygous recessive. The third type of cross that you are going to get, we're going to take homozygous dominant and heterozygous, and we would have very similar results to what we have in 2. Capital B, and capital B, small b, except here you have 50% heterozygous and 50% will be homozygous dominant. In the genotype, in the phenotype, all the offspring are going to look like the dominant characteristic, whereas with 2, here 50% will look like the dominant characteristic and 50% are going to look like the homozygous recessive characteristic. All right, then our 4th one is going to be heterozygous and heterozygous cross. So here we'll have a capital B, heterozygous, capital B, small b, the same here, and here we have small b. So whenever you have a heterozygous and a heterozygous cross, you are going to have 25% will be homozygous dominant, 25% will be homozygous recessive, and then 50% will be heterozygous. But in the phenotype, in other words, what you are going to see, 75%, let's do it like this, 75% of the offspring should look like the dominant characteristic. So whenever you want to do a cross to establish whether something is homozygous dominant or heterozygous, you always make sure that you end up with two heterozygous, put them together, and if there is a homozygous recessive baby that results, or offspring that results, then you know that both parents were heterozygous. So you use what we call a back cross. You start with the offspring and you work backwards. Okay, let's move on. But those are the only 4 crosses that you will ever get. Now, Mendel's law of dominance, please people, it's dominance and segregation. What he says here is that a dominant characteristic will be seen in the phenotype, in other words, the physical appearance, when it's homozygous dominant or when it is heterozygous. You will see the dominant characteristic. And the recessive characteristic will then be covered. It will be shaded because it's recessive, it's not dominant. All right, so he says, here is a spelling error, so let's just fix that. Where 2 individuals with contrasting homozygous characteristics are crossed. The individual of the F1, remember F1 is your first family, it's filial 1, it's the first set of family that the parents get. Hybrid generation will all resemble the parent possessing the dominant characteristic. In other words, if we take a guinea pig with black fur as dominant and a guinea pig with white fur as recessive, when they cross, all the babies are going to be heterozygous. Let's just go back here. If we take a homozygous here, you take a homozygous parent for one trait and a homozygous parent with another trait. So they're both homozygous, but one is dominant, one is recessive. All the offspring are going to resemble the parent with the dominant characteristic. And that is Mendel's first law, or the law of dominance and segregation. Okay, please, you've got to remember that. So, if we look at this entire definition and it goes wow, 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 and you think, oh my goodness, I'm never going to be able to know this, just look at it. It says when 2 individuals, male and female, with contrasting homozygous characteristics, so homozygous dominant, homozygous recessive. They are pure dominant and pure recessive. When they cross, all the babies will resemble the dominant parent, okay? Because they'll all have the dominant gene. They'll all have the dominant characteristic. And that's it. It's as simple as that. So don't get all thrown with the fancy words and whatever. 2 parents with homozygous characteristics, okay, they are homozygous. The offspring in the F1 will resemble the parent with the dominant characteristic. Now, this law shows the principles. I just want to get my pen back here. This law shows the principles of dominance and recessiveness using the characteristic of height. Now, that's what Mendel used. Remember, we're looking at him and his use of garden peas. He used tall garden peas and dwarf garden peas, and they were different species, and he then mixed the 2 together. All right, pea plants grow either tall or, so here they'd be homozygous dominant, here they would be heterozygous, or are short plants. So you have your TT, which is your homozygous recessive, and then they are little dwarf plants. Mendel crossed the tall and the short varieties to prove his theories. So if we look, the F1 offspring show the characteristics from both parents. The F1 generation phenotype, all are tall. The genotype, they are all going to be heterozygous. Okay, now I'm going to just do across, just to remind you here, this is what we are going to have. We're going to have homozygous tall and homozygous short. These are our gametes. All right, we're going to have heterozygous offspring, or 4 chances. So in the phenotype, what are they all going to look like? They all have that tall gene, so they are all going to be tall plants. But what do they have? They have this recessive gene as well. So we're saying here in the genotype, they are all heterozygous. And in the phenotype, the physical appearance, they are all going to look tall. All right, the plants mature and reproduce. Now what Mendel did is he crossed the tall and the short plants. He crossed them. And then he said, okay, let's look at the offspring of the F2 generation. So we have these babies here, who are the F1. They now become the P2, the second parent generation. All right? They mature and reproduce. The gametes for tallness and the gametes for shortness are presented because the gametes segregate into tall, tall, and short, and short. So half the gametes will contain the characteristic for tallness, and half of the gametes will contain the characteristic for shortness. So the other half will contain the characteristic for shortness. I've just said that. I'm repeating myself. The characteristic for shortness is recessive. Remember, it's recessive. That's why it's written with a little letter. Will appear in the second cross offspring called the F2 generation. All right, people, let's look at the P2 generation. So we're looking at P2, and here we are going to have heterozygous plants, because remember, these are the offspring from P1. So our F1 generation now becomes P2. And here we're going to have homozygous dominant, heterozygous, another heterozygous, and 2 homozygous recessive. So if we look at the genotype, we are going to have 1 homozygous, we're going to have 2 heterozygous, and we're going to have 1 homozygous recessive. If we look at the phenotype, all the plants that now possess the T, the capital T, they are going to be tall. So therefore, we're going to have 75% of the offspring are going to be tall, and 25% are going to be short. And that is Mendel's law of dominance and segregation. And every time an organism produces offspring, it will be based on those 4 options. So as human beings, for example, if this was for tallness and shortness genes in us, then we would have 75% chance of our babies being tall and 25% of the baby being short. However, those 4 options will occur every single time reproduction takes place. So every time there is an egg that has to be fertilized by a sperm cell, those 4 options will be there. So, for example, in sex determination, as we did when we did DNA and genetics, DNA and nucleic acids, for your sex determination, every single time a baby is created by fertilization, you are going to find that there is a 50% chance that it'll be male or a 50% chance that it'll be female, every time. So if you are one of 4 brothers, well, guess what? That's highly possible, even though the chances for each conception is 50% boys or 50% girls. All right, now, if we look down at the different examples of dominant and recessive characteristics, I just want to get down here. What have we got? And what I've done is I've given you a couple of examples so that you set for when you get exam questions. But brown eyes generally will dominate over blue eyes, unless they tell you otherwise in an exam. Curly hair generally dominates over straight hair. believe it or not, even though there are quite a lot of people that have straight hair, but there should be a lot more people that have curly hair. And then you've got the semi-curlies and you've got the fizzies and whatever. We're never happy with our hair, aren't we, girls? I mean, that's just a fact of life. But curly hair generally dominates over straight hair. Black hair will dominate over white in animals or blonde in humans. Normal skin pigment, where we have melanin, all right, will dominate over albinism, because albinism is a lack of melanin in the skin. That's why people that are albinos or classified as albinos have almost a white-pink skin. And the freckles that they have result because the skin is damaged. And they'll also have blue eyes because there's a lack of pigment in the irises. Tongue rolling, now believe it or not, I mean, how many of you have ever thought that to do this, I can do it, which means I have the gene. This is dominant over people that can't roll their tongues. Now, I'd like all of you to try. Take your tongue and roll it into a tube and see if you can do that. If you can't, there is a gene that determines that you can't. And I promise you can take a pen, you can try and do whatever you like. You are not going to be able to roll your tongue if you do not have the gene. So, a flat tongue, believe it or not, is recessive. Looped earlobes versus straight earlobes. Now, a looped ear, in fact, you're not going to be able to see if I show you on my face, but if this is the cheek or the side of the face, some people have a straight earlobe and it goes up like that. This would be a straight earlobe, and a looped earlobe would look like this. I mean, these are things we've never ever, or takes a lot. I think if you're a biology learner or a life science learner, you will actually take note of something like this now. But who looks at ears other than to look at earrings or whatever? So this is a looped earlobe. It's got almost a little gap here before it joins the skin, and here it joins the skin straight. And that is a dominant and a recessive gene. Then we've got normal blood clotting versus hemophilia. Now, hemophilia is very important. It's one of the issues that we are going to go through when we look at genetic diseases, because that is a genetic disease that's been passed on. All right, and then A and B blood groups versus O blood group is always recessive, and that we've done when we did blood groups and the different DNA processes to produce the 6 different types of blood groups. All right, now, just to get your juices flowing, we have question 1, and it says, in humans, the allele, let's highlight this, in humans, the allele for blue, look at that, is written with a small letter. Remember, when we did terminologies for genetics last time, what did we do? We said any dominant characteristic is written with a capital letter, and any recessive characteristic is written with a small letter. Try to avoid letters like S's and O's and W's, because it's very difficult to distinguish the differences. But for B, well, it's clear. So blue, they tell you here, little b. So immediately you know it is recessive to the allele for brown eyes, which is going to be dominant. All right, a man, he's heterozygous for brown eyes. So people, immediately, if he is heterozygous for brown eyes, we know that our gentleman, okay, is going to be a capital B, small b. Why? He's heterozygous for brown eyes, marries a woman with, look at this, blue eyes. So our female is going to have blue eyes. She can only have blue eyes in the phenotype if she has 2 recessive genes for that characteristic. So the alleles pair, or the alleles are recessive. Otherwise, she has no blue eyes. Now, I have blue eyes and I have blonde hair. That means for this to be here, although it was blonde, it's now quite a lot gray, but when I was little, it was blonde, all right? And you will find that with you as well. As you get older, your hair gets darker in the phenotype, but your genes still hold the 2 alleles for blonde hair and for the blue eyes. So let's have a look at this. She has to, in order to have that blue-eye trait, it must be 2 recessive genes. Now, show how the possible genotypes and phenotypes and the ratio of the individuals with brown hair and blue eyes in the F1 generation may be obtained. Now, this is 8 marks. So let's put this down. So the first thing you are going to do is you are going to write what the parents are so that you know where to start working. So we say, right, we have the P1, which is the first parent generation. Now, we're going to say we have brown eyes, we have heterozygous brown, which is the father, which is the male, and that is going to be capital B, small b. And we have homozygous blue. So that's going to be small b, small b. Now we know what the mom and the dad are. So we're going to have capital B, small b, heterozygous, times homozygous, which is 2 small b's, your homozygous recessive. Now the first thing that's going to happen is we are going to have meiosis. So let's take our Punnett square. All right, and here we are going to have our gametes. And the gametes are produced by meiosis. All right, very important to put that in. Now we have the father and we have the mother, or the male and the female. Okay, let's do our cross. Capital B, small b, and small b and small b. And we have capital B and we have small b. All right, so this is what we are going to have here. So now, first thing you have to write is, in the F1, what are we going to have? In the, they wanted, remember in the question, they asked us for the genotype, the phenotype, and the ratio. So if you get into the habit of writing it properly every time, it's not a problem. So we say genotype. What do we have in the genotype? In other words, the genetics. What are we going to have genetically, or two possibilities in this cross? We're going to say, right, genotype, we're going to have 2 in 4. There's my ratio. Capital B, small b, which is heterozygous. And we're going to have 2 to 4 are going to be small b, which is homozygous recessive. Okay, now, in our F1, we're going to have the phenotype. People, this is what we are going to see physically. Now, what are we going to see physically here? 50% of the offspring are going to have brown eyes, and 50% of the offspring are going to have blue eyes. Or there are your ratios, your chances. And with this, this is where your, let's do our mark allocations. I'm going to do them in purple. Remember, it's 8 marks, so here are your mark allocations. You're going to get 1 for that, for putting this out. Then you get 1 for this. Then you get, it's 3, 4, 5, 6, 7, 8. And there you have it. Easy, easy 8 marks. And it really wasn't hard. Now remember, I told you there are only 4 types of crosses you can get. You have heterozygous and homozygous recessive, or heterozygous and homozygous dominant. There you go. There are only 4 types of crosses. So make sure that you know those 4 types of crosses and what homozygous and recessive means, what F1 and P1 means, and what your genotype and phenotype is. And if you've got that waxed, I promise you now, you get full marks. For genetic calculations like this, or diagrams, or representing a cross, at the end of the day, you either get full marks or you're going to battle to get marks. If you understand it, it's full marks every single time. So that's easy. Right, now we go to question 2. And I need to make sure that you understand this and that you really, really know this. It's the ability to curl your tongue, I told you just now, to form a tube that is passed from parents to their children. Okay, so we're talking about tongue rolling. Study the diagram below, which shows the inheritance of a tongue rolling in a particular family, and then answer the questions that follow. So let's have a look. This diagram here is called a pedigree chart. And when we look at the pedigree chart, they give you a key, and the key will tell you here, the white circles are female non-rollers, and the males are, your male non-rollers are also not shaded. Your female rollers are shaded and your male rollers are shaded. Now, to roll the tongue is dominant. Okay, it is a dominant characteristic. Okay, so let's just look at our questions and then we're going to go back to our diagram. It says, using capital T to represent the characteristic for tongue rolling, okay, so it's dominant, and the small t for the inability to roll the tongue. Give the phenotypes and genotypes of the individuals. So we're going to go back and we're going to do all these individuals, because this is how you will answer any question related to a pedigree chart. Okay, so you take your pen, let's get our blue because that's a nice contrasting blue, and we say, okay, your female tongue roller is going to be a capital T. They can roll their tongue. And the male is going to be a capital T. We don't know if it's homo or hetero yet, but it's capitals. And your non-roller is always going to be 2 small t's. So let's look at it. For a parent who is a non-roller, so let's go to all our shaded ones. These are all little t, little t, little t, little t. Just bear with me while I fill these in, and you'll see why I'm doing this now, because it makes it so much easier. All right, so all of these are little t's. Now, for parents to produce an offspring that is homozygous recessive, in other words, 2 little t's, what does that tell you? Where did the 2 little t's come from to make that baby? One had to come from the father and one had to come from the mother. If the father didn't have a little t and the mother didn't have a little t, how on earth do you have a baby with 2 little t's? One has to come from the father, one has to come from the mother. So we say, right, have a look here. If A and B produced a baby that has 2 little t's, it automatically tells us that these parents here must each have a little t. Otherwise, they can't produce a baby with 2 little t's. But they are, we know, tongue rollers. So therefore, they must be heterozygous. People, I hope this makes sense. But as we go down now, we're going to do others. But if you're going to produce, if 2 parents are going to produce a baby with the homozygous recessive trait, it means that there must be 2 little or recessive alleles. And if they are recessive alleles, one had to come from the one parent, one from the other. But clearly here in the phenotype, the parents A and B can roll their tongues. Now, if they can do that, it means they must have the dominant genes. But they can't be homozygous dominant, because then the baby couldn't have the 2 recessive genes. I hope this makes sense. So for D, at least for C, D, and H, well, they produce an offspring that isn't able to roll their tongue. Is able to roll their tongue. So we do know that they must have the dominant gene, otherwise they would not be able to roll their tongue. Now let's have a look. These 2, G and H, produce 2 babies. One that doesn't roll their tongue, and one that can roll their tongue and one that doesn't. So what does that tell you? What is T's ability here? T has to have a little, H must have a little t, because we take those two little t's, and the two little t's will then end up producing that little baby there. So they must each have their recessive traits. So we know that H has to be capital T, small t. Now, C and D, they produced a baby who was heterozygous. So they could have been, but one of them must have a small t. Or, so people, with determining C and D, we're not sure whether those parents are homozygous dominant or heterozygous. But one of the parents will have to be heterozygous. Otherwise, where did the T come from for the baby or the offspring H? So we want to make sure that this offspring here there has to be a small t. So we're either going to have capital T to there, or we could have had these two to there. But those are the options for those parents. But we definitely know that this H is a small t, a capital T, small t. Now we go down further and we say, okay, this offspring here can only be heterozygous. We know he has to have a dominant gene, and that if the one parent here, G, is small t, then the only option is a small t. Now going down to this process here, both L and M are produced from I. And if they are, it means that each one must have a little t. So it means that these babies got the dominant gene from the mother and the recessive gene from the father. And if we look at the offspring here, does it tally? Yes, it does, because small t, small t, small t, small t, and small t, small t, which means these babies here could be capital T, or capital T, small t. So both these offspring here can either be dominant or recessive, because we know the parents were both heterozygous. Okay, now let's look at our questions. And the first question says, using T to represent the characteristic and small t the characteristic for the inability to roll, give the genotypes and phenotypes of the individuals A, H, and I. Now the way I would write this in an exam is I would say that A, H, and I. First thing they want is the genotype. So the genotype for A, let's look. A was capital T, H is capital T, small t, capital T, small t, capital T, small t, and I is small t. So we move on here. So we said A was capital T, small t, I was capital T, small t, H and I is small t. Now remember, we've now done the genotype. Let's look at the phenotype. What are we going to be able to see physically? Well, clearly there's a dominant gene, so this would be a tongue roller. And this one here would be a tongue roller. And this one here would be a non-tongue roller. And what they're doing here, your mark allocation is 1, 2, 3, 4, 5, 6, and there you go. What were the chances of I and J's first child being able to roll his tongue? Explain your answer. Now it's I and J, so we go back to our diagram and we look at I and J. And they want to know what the chances are that their children, their offspring, would be able to roll their tongues. Well, look, they are going to be heterozygous. So the offspring are heterozygous. And we say, remember, it's also explain your answer. Don't forget this part of the question, people. So I and J's first child being able to roll their tongue, it would always be 50%. All right? Since there is a 50, why? Now we have to explain it. There is a 50% chance of obtaining a capital T, small t combination. All right, that's what it is. You could also say there's a 50% chance of a small t combination, so therefore your capital T, small t will always be 50%. And there you've explained it, and you get your 2 marks. Why is the character for tongue rolling given a capital letter rather than the character for the inability to roll the tongue? Use the information from your diagram to answer your question. Now, all we have to, our diagram was one back. All we have to do is when we look at this diagram here, apart from the fact that you can't say, well, the key says so. That's not an answer. What we do here is we look at the fact that tongue rolling is always dominant. That's why it's the characteristic for written with a capital letter. If I have here heterozygous producing a homozygous, and here again, homozygous, recessive producing a homozygous baby, here you've got homozygous and heterozygous, and you end up with a child who cannot roll their tongue. So from this diagram, you would then answer, why is the character for tongue rolling given in capital letters? Because it is the dominant characteristic. Okay? And so tongue rolling is the dominant characteristic. And why? Because the ability to roll the tongue is dominant. Okay, so for tongue rolling, then you will see, let's just put this up a little bit. You will see on the diagram that L and M have 5 children, 2 of whom are tongue rollers. So out of 5 children, 2. So 2 out of 5 can roll tongues. And do you see what I'm doing? I'm writing on my question paper. So 2 of those 5 children can roll their tongues. What are the chances of the next child being able to roll his or her tongue? So let's go back to our diagram. Sorry. Here are L and M, and they have 5 children already, of which 2 can roll and 3 can't. So we say, okay, what are the chances that their next child will be, now we've got capital T, small t, and the 2 of them, it goes the same every single time. Remember, every time a child is produced through fertilization, or an offspring is produced through fertilization, those 4 combinations will apply every single time. So we're going to do the cross here. We say the 2 parents are heterozygous and heterozygous. So capital T, capital T, capital T, small t, capital T, small t, and small t, small t. So what are the chances that they're going to produce children that can roll their tongues? It'll be 75%. What are the chances that they'll produce a child that can't roll their tongue? Will be 25%. So the answer to our question, 2.4, will be, what are the chances that the next child can roll their tongue? It will be 75%. Why? Because you've got a heterozygous crossed with a heterozygous. In your phenotype, it'll always be 75%. 25% homozygous, dominant and 50% heterozygous, and the two together, what are you going to see in the phenotype? 75% can roll their tongues. All right, now, you will see that P can roll her tongue. What are the chances of her being heterozygous for this condition? Explain your answer. So we're now looking at P. P are able to roll her, is able to roll her tongue. So what are the chances that she's heterozygous? It will be 50%. What are the chances that she's homozygous dominant? 25, because she is either capital T small t or small t small t. So there is a 50% chance that she is going to be heterozygous. You see this, people? 50%, because there it is. That and this. In fact, let's do it like this. There. Is that P is going to be able to roll her tongue as a heterozygous organism. All right. Now we get to Mendel's second law. And this second law is very important. What the second law says is that each characteristic and each gene for a characteristic separates independently. In other words, now think of the people that are around you. You can have people in one family where the mother has dark hair and she has blue eyes. The father has brown hair and he has brown eyes. They will produce children who have blonde hair, brown eyes, and let's throw skin in as well, so maybe a very dark skin. Or you would have a parent who has a child with curly hair and the parents both have straight hair. Do you follow what I'm saying, people? There are a lot of combinations. And if you have dark hair, it does not mean that you will automatically have dark eyes and a dark skin. Or if you have light hair, that you will have light eyes and a very pale skin. All these combinations means that as the organism is produced, is made, those genes segregate out, and when fertilization takes place, you can have a variety of combinations. So you can have a person with a dark dominant colored skin, with a recessive, homozygous recessive hair, and a heterozygous set of eye colors. So they segregate independently, each characteristic. So he said that each characteristic is controlled by genes. And they are going to be your allele genes. Now, those allele genes are located on, look at this, a separate pair of chromosomes. They are completely separate. So therefore, every combination that takes place to produce a new organism is going to be separate. So Mendel stated that the different pairs of chromosomes segregate independently. People, this is incredibly important. They segregate independently every single time in a hybrid cross. Now, this means that each characteristic is determined separately from the others. All right, completely separately, so that we have a combination that is separate. So here we've got the separate characteristics. They are independent of the other characteristics. That is why you can have blonde hair and brown eyes and a very fair skin, or you can have dark brown hair, dark brown eyes, and a very fair skin, or whatever the combinations are. They are independent, so we have independent assortment. So Mendel used his little peas again. You see, he liked his peas. And he chose different types of peas. He chose peas that were round and wrinkled, the seeds, and he chose seeds that had a yellow color and some have a green color. Now, possibly your teachers will go and take money and buy a packet of sweet pea seeds. They're actually the perfect time to plant sweet pea seeds is autumn time. so that your sweet peas flower in winter, and they actually are beautiful. So if you have a classroom or even at home, if you have a place that gets a lot of sunlight in winter and is nice and sunny, go and get yourself a packet of sweet peas and plant them and watch how they grow. But when you look at the seeds, before you plant them, have a look at those seeds. You'll see some are all wrinkled and scrunched up, and others are perfectly round. And you'll see some are a darker shade of green, and others are more a mustardy, yellowy color. And that is what Mendel used. So you can go and do Mendel's whole process here. Go plant your little sweet pea seeds, and then 2-3 months later, you'll have the most wonderful smelling garden. In fact, if you plant it near your bedroom window, you're going to have wonderful smells coming up. All right, a genetic trait for round seeds versus wrinkled seeds. So we're looking at round seeds versus wrinkled seeds, and we're looking at yellow versus green color seeds. So we're looking at 2 characteristics. Now, what kind of combinations are we going to have here? This means that we can have the following. We can have seeds that are round and yellow. Because remember, we've got 2 lots of characteristics. We've got round, we've got wrinkled, we've got green, and we have yellow. So we can have any of those groupings together. So they can be round and yellow, they can be round and green, they can be wrinkled and yellow, they can be wrinkled and green. So at the end of the day, we are going to have 16 combinations, wherein our normal monohybrid cross, 1 characteristic cross, we are crossing 1 characteristic. There are always 4 options. There are 4 combinations of the genes. Why? Because we've got 2 gametes from the mom, 2 gametes from the dad, and that gives us 4 different combinations. Here, we are going to have 16 different combinations. Why? Because we've got 4 times 4. So it's the 4 for the 1 characteristic and 4 for the other characteristic multiplied together. So 16 different types of groupings. And this can be mind-boggling. So there are certain rules here that I'm going to explain to you as we go so that you do not get confused. But if you are asked to do a dihybrid cross in the exam, you have to keep your wits about you and pick 1 characteristic, and that is your lead characteristic. So when you write all your symbols down and all your letters in your blocks, make sure that you keep the same letter in the front. So here we go. I'll explain it to you as we go. Your dihybrid, so dihybrid, di, remember, di means 2. Okay, di and bi means 2. So dihybrid cross, we're going to have 2 traits or 2 characteristics that we are going to be crossing. Now, homozygous parents. Now, the minute they're homozygous parents, we reflect back to when I said to you there are only 4 different types of crosses. If you have homozygous recessive versus homozygous dominant, and the crosses will all be heterozygous every single time. Here we go. We have the capital R for round seeds Y, that is dominant, and a capital Y for yellow seeds because it is dominant. So yellow dominates over green and round dominates over wrinkled. So here we're going to have the small r because it is recessive and the little y because it is recessive. Now, if your handwriting is not very neat, you're going to battle with the capital Y and the small y. So people, be very, very careful the way you write this down. Be neat. First step. This is step number 1 because there are 2 main steps. Step number 1, you are going to have a parent, all right, that is going to be homozygous for the dominant characteristics and at least homozygous for the recessive characteristics. So that will be recessive, and this is going to be dominant. So homozygous dominant and homozygous recessive. Okay, now we've got these parents, but now we have to find the combinations of the gene, at least of the gametes, that each parent's going to produce. So step one, you look at the combination of the parents, and then you have to split those gametes up. So we say, right, for this parent here, this is parent 1, that's parent 2. So for parent 1, which was capital R, capital Y, we're going to now do a genetic cross. These are our gametes that we are going to end up with after meiosis has taken place. So capital R and Y, capital RY, capital RY, capital RY. So that's parent 1's combinations. Parent 2's combinations, we are looking at little r and little y. So we do exactly the same cross here. Meiosis occurs to form those gametes, and these are our groupings, our results. Now, once we've got this, we now move on to the second step. This is step 2 of our dihybrid cross. If you just keep your wits about you, you must first find the gamete combinations. Now we've got them. Now we can move on to putting it into our Punnett square. People, 16 combinations. Now, if you remember if we only had one characteristic, we had here, let's say, capital R, small r, and capital R, small r. We had our 4 different types of combinations, so there were 4. Here, we have 16. There's 1, 2, 3, 4 times 1, 2, 3, 4. So we're looking at 16 different combinations. Right. Our dihybrid cross, there's our first parents. Remember, the parents were capital R, capital Y, and small r, small y, small y. So now we cross them. Pick one of your traits. So you pick one trait, and you look at the dominant trait. And that will be the one that you go with. So in this case, we chose R. So R would be our letter. So anything that had a capital R or has a small r, if it was a different type of cross, we would keep R in the front and Y at the back. If you look here, all the R's are in the front and the Y's are all afterwards. So capital R, capital Y, small y, Y. I've taken a homozygous and I've crossed them. You will always end up with heterozygous. So 16 out of 16 offspring in the genotype are going to be heterozygous, capital R, capital Y, small y. In your phenotype, 100% of these babies, remember round was dominant and yellow is dominant. So they all contain the dominant gene. All the offspring are going to be round and yellow. The fun part comes in now when we cross the offspring and the F1 generation becomes P2, becomes the second parent generation. And here we have our fun and games. Now we go to P2 and we say there's our heterozygous male and our heterozygous female, and we are now going to first find what each of the parents are going to, their gametes that they're going to have. So the parents, this will be parent 1, this is parent 2, and the 2 now split up. The gametes, we're going to have meiosis, and we're going to have capital R, small r versus capital Y, small y. So 50% are going to be homozygous dominant, and 50% are going to be at least 25 are homozygous recessive and homozygous recessive. So same thing over here, they're going to be exactly the same because the parents' gametes are the same. Now we've got our 4 combinations for the one parent and we have our 4 combinations for the other parent. So we move on. With our gametes here, there's our capital R, capital Y, so that would be our dominant combination. Here round would be dominant, but the color would be green. Okay, here we're going to have a wrinkled little seed, but it's going to be yellow. And here we have a wrinkled little seed that is green. So these are our combinations. So we've got heterozygous versus heterozygous. Now, our F2 generation people F2, because this is P2, F2 occurs here now. And we say, right, what do we have? The gametes undergo meiosis, and then we have fertilization. Now, as fertilization occurs, what do we result with? When you write these out, make sure that you pick your one letter. As I said, in this case, we chose R. So there would be capital R's. There my R comes for always capital R, and then if they're small r's, then you write them second, or little r, little r. But we keep R in front for all the combinations. And then Y, if they're both capitals, that's great. If there's a capital and a small, we keep the capital first and the small last. So your basic rule with a dihybrid cross, you are going to pick one of the traits, and that will be your leading trait for all your combinations. In this case, we chose R, the capital R, for round. So we chose round, the shape, and then we chose the color. So R would be first, and if it's a capital, you write the capital first and then the small letter. But you stick to round being the dominant, and then you put the color. So first the shape and then the color for all your combinations. Otherwise, look at this. You end up with something like this, where you now, for example, here you have Y, Y, capital R, small r. And here you have capital R, capital Y, small r, small y, and you look at this, and then some are r's and some are y's. You're not going to be able to see what your combinations are for your genotype. So make sure the same letters in the front for all of them versus the second characteristic. So you pick your characteristic, okay? You pick your characteristic every single time, and you make sure that you stick to that same characteristic, and you write the capital first and then the small letter. And if you keep to that rule, you can't get confused. So our combinations for our genotype, 1 in 16 will be capital R, capital Y. So homozygous dominant for both traits. 2 in 16 will be round and yellow. We've still got here 1 in 16 homozygous round, homozygous yellow, and so we green at least, and so we carry on. So you write all these combinations out, everything that you have here, and as you write them down, you cross them. So capital R, small, capital Y, cross it out, and you've now written it down. Capital R, capital Y, small y. You've done this one, and you've done that one, so you tick it. Our next one that they wrote down here was capital R, small y. There's that one, and you've done it. And this is the only way to check. You look at it, write it down, and cross it out. So cross out, cross out. There we've got our 2. And cross out, and there should be another one. There we go. We have our 4. That is the only way to keep track that you've done it properly. Cross them out as you go on all the way through. All right, now, phenotype. Remember, round is dominant, and we're going to have yellow is going to be dominant. Okay, so anything that has a capital letter in it is going to be, that is what you're going to see. So we're going to have 9 round and yellow. We're going to end up with 3 round and green. We're going to end up with 3 that are wrinkled and yellow. And here is our little recessive, homozygous recessive. We're going to have 1 that is wrinkled and green. And this is how you're going to do it. We say that this is a 9 to 3 to 3 to 1 ratio. And you will always have this 9 to 3 to 3 to 1 ratio when you have 2 heterozygous parents. All right, so when the characteristics are heterozygous. And I'm hoping you've grasped this. Focus on your monohybrid crosses, then your dihybrid crosses are just an extension of it. All right, if we look at question 3, what do we have? We're saying, with reference to the hair coat in a guinea pig, black is dominant over white, so that's important. Black is dominant, White is recessive. That's why black is written with a capital letter and white is written with a small letter. And rough coat is dominant over smooth coat. Okay? Provide the phenotypic or phenotype ratio for the F1 in the following crosses. So here they're saying we're looking at the first family, the first offspring for this cross. Now, before we go into this whole question process, I'm going to recap. We're going to go back and we're going to look at Mendel's law of independent assortment. Again, just to refresh your memories. Okay, look at this again. Each characteristic is controlled by allele genes. We know this. Allele genes are located on separate pairs of chromosomes. Mendel stated that different pairs of chromosomes segregate independently. Okay? Segregate independently. So they are not dependent on one another. This means that each characteristic is determined separately and that each combination will be separate and independent. And that is why we do step 1 in a dihybrid cross, because we're looking at what the combinations are from the one parent and what the combinations will be from the other parent with their different groupings or types of traits. All right? We looked at his P's and our combinations. We are always going to have 16 combinations in a dihybrid cross. Now, if you've got 4 combinations in a monohybrid cross, and a dihybrid is double the characteristics, it's going to be 4 times 4 is 16. Okay, so having done this, let's go to our question. And you're going to do this with me. So, capital B, small r, and small b, capital R, capital R. We're going to look at these parents here, and we're going to look at what they are going to produce. So we say this is parent 1, and that's parent 2. So parent 1, just for our own writing out on our page, we're going to say this parent is going to be homozygous black, and it's going to have little r, so it's going to be homozygous smooth. All right, and then our second parent here is going to be homozygous white. So homozygous white. They're completely opposite, these organisms. These are 2 little guinea pigs. They say opposites attract, and this one's got a rough fur, so it's going to be homozygous rough. All right, so we've got a black smooth-haired guinea pig, and we have a white rough-haired guinea pig. A little bit of a disheveled look, maybe puts a little bit of hair glue or something, you know, the paste stuff, and makes it look all spazzy. We're now going to do our cross. And this cross is going to be lovely because we're going to say, we've got a step 1, so step, and please do this for yourselves. It's not going to give you a mark to write step 1, but it helps you to focus on where you're at, and so that you don't lose your place. So step 1, we are now going to find what our combinations are. So we say, we've got parent 1 is going to be capital B, and small r, small r. We have our gametes here, and our gametes undergo meiosis. So we're going to have capital B, small r, capital B, small r, and guess what? The same thing here because it's homozygous. And for our second parent, we have our gametes, and we have meiosis taking place, and our meiosis is going to result in a little b, and capital R, capital R. So now if I chose B in the front here, I have to do the same here. So B, capital R, B, capital R, B, capital R, and B, capital R. Now we are ready. So we've now done our first step. We now know what our 4 times 4 combinations are going to be. Okay, now we go. And I'm not using rulers because I want you to see how to do it. Okay, this is what is important. So, looking at all our combinations, we know we are going to have our gametes. Our gametes are going to undergo meiosis, and these are our combinations. We're going to have capital B, capital R, capital B, capital R, capital B, capital R, and capital B, small r. And here we have this. And we have it all across. Here we're going to have fertilization occurring. And if fertilization occurs, these are our combinations. So in every single case here, we are going to have capital B, small b, capital R, small r. And they are all the same combinations. So we are going to have, these are, remember, our P1. So F1, what do we have? In the genotype, in other words, what is in the genes? We are going to have, remember, they're all going to be capital B, capital R, small r, all of them. So the genotype, we're going to have 16 to 16 will be capital B, capital R, small r. So they are going to be heterozygous. Okay, and our phenotype, what are we going to be able to see? 100% of the offspring, 100% are going to be our characteristics. We go up and we say black and smooth and white and rough. So our babies are going to be black and rough and have rough hair, rough coat. 100% of them. And that is what we will see. And they will all be heterozygous. All right? Now, if we go back to our question, and it said here, with the reference to the hair coat, etc., we now have a second combination because they said we have to do the F1 for the following crosses. Here we've got capital B, capital R, small r, and we're going to have capital B, capital R, small r. In other words, we are going to have heterozygous organisms. They are going to be heterozygous for color, and they are going to be heterozygous for rough and smooth hair. So when we look at these little guinea pigs, they are going to be black, and both of them, they're both going to be black, and they are both going to have rough hair. Because look here, your capital R is for rough, and there is our capital R, and it's the dominant gene. And color, black is dominant. So we now go all the way to a new page, and we say, or let's just do it on here. We're going to have our combination, capital B, capital R, small r, times capital B, capital R, small r. These are our two organisms. There is the female and the male, or the other way around. Parent 1, parent 2. Step number 1, first step, step one, what are we going to do? We're going to say, what are the gene combinations? So our gene combination for the first parent, I'm just going to put your gametes, and they're going to undergo meiosis. And what do we end up with? Capital B, small b, capital R, small r. Now remember, they are identical, so therefore this Punnett square will suffice for both of the parents. So we decide to put B first, so we go capital B, capital R, and we're going to have small b, capital R. Why do I put small b first? Because I've decided to go with B's. Then capital B, little r, and small b, small r. And this would be the same for this as well as that parent. Now, we go P1 and our P1. This is the combination set we are going to have. And then we go our gametes, and the gametes undergo meiosis, and these would be all the combinations we have when fertilization occurs. All right, now keep your head about you. We're going to have capital B, capital R, small b, capital R, capital B, small r, or small b, small r. I've just gone the top two, and then I've gone bottom two. It doesn't matter what order you do these, write these in. So if you decide that that is 1 and this is 2 and this is 3 and that's 4, it doesn't matter. Just remember, your B's must be in front for all of them, because that's what we chose. If you select R, that's great too. But once you've made that decision, keep to it. And we're going to have exactly the same down this side. We'll have capital R, capital B, capital R, small b, capital R, capital B, small r, and small b, small r. Now, when we write these combinations in, we have to keep our heads about us. So we say, right, we have capital B, capital B, capital R, capital R. What have we got though, people? We have a heterozygous cross. We know from experience it has to be a 9 to 3 to 3 to 1 combination. Okay, whenever there's heterozygous and heterozygous, just go up here. We've got heterozygous and heterozygous. It must be a 9 to 3 to 3 to 1. So that's easy because that helps us. So there's one of them. Then our next combination, we go along this way, and when we finish that row, we move on to the next row. So here we're going to have a capital B, a little b, and 2 capital R's. And here, capital B, and a capital R and a little r. And a capital B, small b, capital R, little r. Across this way. So here we go, capital B. Remember, our capital's always first, and the small letters second. So capital B, small b, R, R. And we have small b, capital R, and I'm not going to say R, R, B, B, just watch what I'm doing, because it gets a little monotonous when I say each of these letters. Maybe you can hum, sing, if you've got a singing voice, while we do this, but it's actually fun. And just concentrate to make sure that you're getting your letters in the right place. Capitals first, small letters second, and your B's always in front. And there's our little one that's going to be homozygous recessive for both traits. So here we have our whole process, and now we have to make sure that when we write down, I'm going to move this up just a touch, when we now look at our F1 generation, we're going to look at our genotype, and then we're going to look at the phenotype. So genotypically, what do we have? Now remember, with your genotype, you have to write down what your combinations are, okay? And we're going to say, right, genotype, the ratios. I'm going to have here capital B, capital B, capital R, capital R. So I have 1 in 16 will be capital B, capital B, capital R, capital R. Okay, then cross it out. The next one is this. So here I've got 2 in 16 will be capital B, small b, capital R, and cross them off. The next, we can go along here. Let's do this one. We've got 1, 2 of them. So 2 in 16, we're going to have capital B, R, and R. So cross them off because we've done them now. They're out of the way. Let's do this little one in the middle here. There's only one. Now all you need to do is run your eyes along and see where you've got 2 small b's, but there's 2 capital R's and a small. So there's only one of these. So 1 to 16 is going to be little b, capital R, capital R. All right, I'm going to write the rest on the side so that we don't end up with too much, or mind you, we could, it's fine. All right, now we're going to go here. We've got capital B, capital R, small r. So capital B, capital R, small r. 1, 2, 3, 4. So it's 4 in 16 will look like that. Okay, next one. We take these. So we go small b, capital R, small r. And there's 1, 2. Okay, and then we do this one. I'm going to write it here. Capital B, small r. We only have 1 in 16. And then capital B, small r. There's 1, 2. So we have 2 in 16. And finally, our poor little one, little homozygous recessive for everything, little b, little r. Now, people remember capital B was for black, and small b was for white, and capital R was for rough fur, and the small r is for smooth fur. Now, when we do the phenotype, and it's not allowing me to move on, but for your phenotype, we're going to put it in here on the same page. I'll do it in a different color. With the phenotype, here we've got black, and black, and black, and black, and black, and black. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 of these organisms, 12 of these offspring, chances, 12 are going to, it's 12 out of 16, are going to have black fur. All right, 12 of them. Of those 12 that have black fur, let's see how many are going to have a rough fur coat. So 1, 2, 3, 4. So that gives us 1, 2, 3, 4, 5, and 6, 7, 8, 9. 9 out of 16 of the black ones are going to be black and a rough coat. All right? Then we have to look at those that are going to be black with a smooth coat. So black with a smooth coat, there's 1, there's 2. So that's 6. 6 out of 16 are going to be black with a smooth coat. And we then look at white, we've only got 1, 2, 3. So 1, 2, 3, 4. So 4 out of 16 are going to be white. But of those 4 out of 16 with white coat, we're going to have 6. Have I got this right? No. There's 3 out of 16 are going to have a rough coat. And we're going to have only 1 out of 16 is going to have a white coat that is smooth. So when we've added all of that together, our results are going to be a 9 to 3 to 3 to 1 ratio. All right? Every single time. Why? Because we have a heterozygous plus a heterozygous. And if you remember with our 4 combinations, so where we only have in our monohybrid cross, so this is di. In our monohybrid cross, if we have a hetero plus a hetero, we are going to end up always with a 2 to 1 to 1 ratio. So just bear this in mind. Your hetero will always be that, where 1 will be homozygous recessive, and 2 of them will be, at least 1 will be homozygous dominant, and these 2 will have your dominant characteristic. Okay, people, that's as far as we're going to go for today. We're then going to move on in the next time we see you. We're going to move on to our genetic diseases and issues that we have there. All right, so I hope you have a good week. Bye-bye.